Okay. So as we have already heard this morning, today, this is the 10th anniversary of the Cholangiocarcinoma Foundation Conference. So we thought it was important that we would stop and just look back at how much our knowledge on cholangiocarcinoma has changed and grown over the last 10 years from a basic research perspective, and how much this has impacted on the clinical management of our patients. So it is my absolute pleasure to invite the next speaker, who has actually played a major role in the advancement of the knowledge of cholangiocarcinoma, which has really impacted all the patients in the future years. Nabeel Bardeesy is a faculty member of the Harvard Medical School and Massachusetts General Hospital Cancer Center, and where he also holds the Gallagher Chair in Gastrointestinal Cancer Research. And he will just give us an overview of what has changed in the last 10 years in basic research. Thank you.
Thanks so much. It was really a pleasure to have the opportunity to review what's gone on in the last 10 years. I have to say that this conference is unique is it allows us to come together, the one place that we can come together with the clinicians, scientists, laboratory researchers who really are focused on this disease, and to be able to meet one-on-one, spend a few days with them is really an incredible opportunity for collaboration and growth. I will say that the most important deliverables, of course, is patient improvements in patient outcome. And we've divided the summary of this conference into two parts, and I'm going to cover the basic science element. So the way I view this, or one analogy, we've heard some analogy this morning, we can think of a car analogy for perhaps cancer research. First of all, we need to know the components. Second of all, we needed to know how the components are assembled, some sort of assembly diagram. And then third, how they actually work together, what makes it stop and start, and so on. And for cancer, we can think of the components as the genetic changes, the gene mutations that are very different between individuals and can derive different biology. The second aspect is how the genes talk to each other, what they turn on or off in terms of other genes. And how things work is how ultimately cancers evade the immune system, how genetic changes cause cancer cells to move about in the body, and then, of course, ultimately, what are the vulnerabilities? What are the best ways to kill the cancer cells? And so I think as I was reviewing the 10 years of this wonderful meeting, I saw a progression from that exact set of metaphors. What are the building blocks? Going all the way to now an emerging view of how things work. And so with that, I'll go to the next slide, please. Okay, so here we go. And again, I've just chosen work that I thought was illustrative of some of the advances. There's lots of terrific work that I won't be able to talk about today, and it's going to be in a very brief format. Again, just to give a sense of the progression and where we are and what we need to do next. So some of the important building blocks or components that we needed to know about related to the gene mutations were work that was presented early on in the early days of this conference by Laura Wood from Hopkins, showing that there's an unusually frequent type of mutation in cholangiocarcinoma in genes that regulate what we call epigenetic states. And these include BAP1, ARID1A, and PBRM1. So this was very valuable because historically, cholangiocarcinoma had been lumped in with pancreatic cancer in terms of the way to treat this disease. And these mutations are exceedingly rare, particularly the first and last in pancreatic cancer. So an indication that we were dealing with a very different entity. Another key advance, and Daniela Sia, who's here today, is one of the key contributors in the area, as well as Arul Chanayan, Mitesh Borad, was that, again, a particularly common alteration in cholangiocarcinoma is of the FGF receptor, which is abnormally functioning because of a break in the chromosome. And this, of course, has led to some very important therapeutics in patients who have these translocations. And again, this work was presented in the early days of the conference. In fact, just as, of course, it informed future clinical trials with FGF receptor inhibitors. Of course, it's important to understand the entire atlas of changes across large patient cohorts because the disease, it turns out, is quite heterogeneous or variable between individuals. And so we had the good fortune of many close partners in the Cholangiocarcinoma Foundation who contributed to the so-called Cancer Genome Atlas project focusing on cholangiocarcinoma, and work presented by Lewis Roberts and Larry Kwong gave an initial view of a more broad landscape of all the different changes that we see across patient subsets. So again, this is giving us the initial assembly, let's say, of the disease at the genetic level. This has progressed now. Of course, the disease isn't necessarily the same across the world. And in fact, in parts of the world, the incidence is more common, particularly parts of Asia. And so important work that was presented here and partly funded by the foundation and other partners from the Dr. Budhu, as well as Bin Tay from Singapore and from NIH, showed that the genetics is quite different. And I'll just point at two mutations. P53 mutations are more common in cancers from Asia, and IDH are less common. And this is particularly true in patients who have risk factors associated with various inflammatory states. So again, telling us the disease is even more different between individuals, although it's still biliary tract cancer. And work again by important partners in the foundation, Jesper Andersen and Lewis Roberts, showed that in fact, risk factors among North American patients and European patients can, again, contribute to shifting the spectrum of gene mutations. And again, with P53 being, let me see, I'll dispense with this, but anyway, with P53 being particularly common in HBV-associated cholangiocarcinoma, and IDH mutants rare in that subset. Again, giving us a sense that we need to kind of really understand subtypes more because there's so much genetic variation. And now in 2023, we have a picture here. Just in brief, I've highlighted some of the sort of salient points that you can see. The enriched mutations in European and American patients. This is not ethnicity, this is probably just geography. IDH, FGFR, for example, whereas in Asian patients and HPV-associated disease, there's other mutations. KRAS and P53 are much more common. We also know now, and that was not appreciated at the early days of this foundation, that extrahepatic cholangiocarcinoma is, again, a different entity genetically, but in fact does have quite a considerable overlap between intrahepatic cholangiocarcinoma associated with these various inflammatory cues. And again, important work by many people associated with the foundation helped further resolve that associations. But this is only the beginning. I had described this metaphor of, let's say, the components. Now, how do the components relate to each other? So now studies have been able to look at subtypes. Mutations in one gene are more frequently associated with another, and this might have clinical implications, and there's a number of studies relating to that. This is far too small to see, but genes aren't the only component. There's also immune cells. Genes give rise to proteins, and so there's so-called multi-omics studies, and this is a very nice one from China, again, trying to take into account all of the different features of a tumor to try to get a comprehensive picture of what has gone wrong, and again, how to mobilize these changes in terms of therapy. Important work by Mark Yarchuan and a number of other colleagues in this room has more specifically focused on how a certain mutation is associated with a certain immunological change. So the cancers evade the immune system. The immune system should recognize the tumor as a foreign entity, but cancers have different strategies, let's say, of evading this. But again, within cholangiocarcinoma, what's apparent is that different genetic subtypes have entirely different properties of how they evade the immune system. And so I just wanted to summarize for where we are now. There's so much variation that I don't think we have a complete picture of the functional subtypes of cholangiocarcinoma. And this is important, I think, for ultimately designing personalized medicines. So I think we need to continue these kind of big analysis to greater numbers of patients, but also to use experimental studies so that we can understand not just correlations, but causations. If a certain mutation is associated with more or fewer immune cells, to actually demonstrate this in a model system, and again, as a path forward for treatment. Other areas that haven't been explored so much genetically, unfortunately, this cancer in many patients metastasizes around the body. There's been far fewer studies in the biology of those tumors. Secondly, we know that the genetics changes upon treatment with chemotherapy or other types of therapy. And so we don't have a comprehensive picture of this. And again, to provide these next generation treatments after, if patients happen to progress on these therapies. Also within tumors, it's possible that there are multiple individual tumor regions that actually quite differ in their properties, and that can also influence therapy. We have methods, I won't go into it now, that have just developed in the last 3 or 4 years, that allows a much higher resolution of how cells talk to each other, something called spatial transcriptomics, and maybe, again, taking into account the broader features of these tumors. Cancers require nutrients to grow, and the way they use these is often unique to genetic context. And so if we integrate the metabolism of the tumor, this might, again, further resolve subtypes. Another key concept here, this might sound a bit more obscure, but I think I'll hopefully try to relate it to therapeutics, is the idea that there is plasticity in the liver. So this is a more basic science question, how the two main cell types in the liver relate to each other, hepatocytes and bile ducts. And very important work from Merrill and Ben Stanger, who's the new Scientific Advisory Board director, has really given us a clue to how cells within the liver actually can change identity. This is the normal or injured liver, not the cancerous liver. And work by a postdoc in my laboratory who sadly passed away recently, Supriya Saha, showed that one of the mutations that happens in about 25% of patients, IDH mutations, plays a role in the ability of injured cells to return to a normal state. And I'll show in a moment, we think that this has significance in the context of cancer as well. Very important work that was presented at this conference by Stuart Forbes, by Luke Bolter, and by Willenbring showed that very clearly that there are mechanisms that underlie the interconversion of liver cell types. And then another recipient of funding from the foundation, Waleski, further resolved these properties. And then in the more direct cancer setting, Aram Hiesel presented on the ability of different gene mutations to cause cholangiocarcinoma when targeting either the hepatocytes or the bile ducts in experimental model systems, telling us that it's very important to understand what it is that allows liver cell types to interconvert. So where are we today? So we have an understanding that in the adult liver, there is capacity for cell types to interconvert, and also for various gene mutations to cause what we call cell transformation or initiate cancer by converting these cell types. We also have evidence that the relationship between the cholangiocarcinoma and a bile duct cell and hepatocyte cell seems to be somewhat dependent on the gene mutations that gave rise to the cancer. And so it's emerging that certain mutations confer a cholangiocarcinoma phenotype that seems to sort of resemble more a intermediate between hepatocytes and bile ducts, whereas other mutations give rise to much more bile duct-like properties of the cancer. Now, this seems very sort of theoretical, but some important work that emerged from Servier, which is the drug company that developed the first approved IDH inhibitor, they had samples where there were pre-treatment biopsy samples and samples of patients during treatment. And they found that the strongest predictor so far, or the strongest correlate for long-term benefit within the tumor, is the upregulation of genes indicating a sort of reprogramming of the cancer cell back to a normal hepatocyte-like or normal-like features of the tumor. So this suggests, again, it's a correlation, but that the subversion of differentiation that was being studied in these basic systems might be relevant to the benefit of therapy. And so where are we right now? So while, for example, in the IDH setting, differentiation correlates with outcome, is it required? Is there a functional role, or is it just simply a correlation? And if it is, what are the mechanisms? And so hopefully we can look at this and maybe exploit further differentiation. Do these other subsets in which there are apparent differentiation blocks, is this relevant to the tumor biology? Can we further exploit this differentiation therapeutically by discovering new mechanisms that support these states outside the gene mutations? And there's another form of plasticity a lot of people work on in cancer called epithelial mesenchymal transition, that in other cancer types really influences the responses to therapy. And this is an area that I think warrants further exploration. I think I'm running somewhat low on time, so I'm going to speed up unless I get instructions otherwise. I have five slides left. Okay, all right. So what do we know about oncogenic pathways? So what do we know about what these cancer genes do? And I've just highlighted a couple of studies here. Again, the work of Bolter and Forbes focused on two pathways that we think are particularly important, biochemical pathways, the Wnt and Notch pathways. In our lab, we've been working on the mechanisms of mutant IDH to hopefully try to devise strategies to make IDH inhibition work. And again, this is probably too complicated for the current context here, but what we found is that a very important role, and the work was presented by Wu and Shi at this conference a few years ago, is that a very important function of this mutation that's in the cancer cells is to cause mechanisms for the tumor to completely evade the immune system to attack the tumor. And it's allowed us to come up with potential strategies that might allow us to better take advantage of IDH inhibition, now that we understand that a very important key is the partial but not complete restoration of immune function. And again, just one more thing from our lab relating to FGF receptor fusions in cholangiocarcinoma. Again, it was work presented by Wu at this meeting, which relates to the mechanisms by which FGFR fusions cause growth of cancer cells, and why FGFR inhibition, or perhaps how to improve upon the impact of FGF receptor inhibition. And just in brief, the work showed that in many models of this cancer, we don't know in patients, but in many models, the inhibitors that turn off FGFR do not completely turn off the ultimate biochemical pathway that FGFR regulates, because there's something called an adaptive signaling program involving another protein called EGF receptor. And by observing this interplay, we were able to, again, only in models so far, devise a strategy that if this is tumor size in the model system, this is untreated tumors, this is the FGF receptor treated, so slowing but no shrinkage, and then the combination with an inhibitor of this other protein called EGFR led to a much more prominent response. And again, this is a hypothesis that can be tested in the clinic. Again, I'll just go over this very briefly here. One very important advance has been the development of inhibitors by pharmaceutical companies that target another common mutation called KRAS, which is very commonly mutated in particularly patients in Asia, but it's also one of the most frequent mutations in patients in this country as well. And now there's an inhibitor, so a drug that turns off this mutant protein very specifically and strongly. And so this is data from Mirati, the drug company, and showed that they had one cholangiocarcinoma model here, and there was, again, early shrinkage. So again, this is very promising because in the past, these patients would not have had any, patients who had these mutations would not have had any selective therapeutics. And then we can use new approaches to identify new pathways, which is technologies which I'll be speaking about more tomorrow, called paralog screening. So it's a new kind of genetic approach that can nominate new targets. I am going to go quite over because I have 3 slides left. So I'll just, again, I think a really important emerging area of the last 3 years was to, again, appreciate that cancers aren't only tumor cells, but an interplay between all kinds of other cells from the body that don't harbor mutations. the blood vessels, the immune cells, and so-called fibroblasts or connective tissue cells. And that there's further potential communication between the so-called microbiome or bacteria in our intestines, which can communicate and send out signals to other parts of the body. And so we've seen important talks from Tim Gretton and Zhang at this conference, showing that eradicating the gut microbiome can influence the growth of these tumors. Important work by Heichenwalder that was presented here that described how inflammation in the liver can influence the initiation of tumors. And a recent awardee and collaboration with Katie Kelly has now really tried to dissect out the different types of immune cells that are associated with cholangiocarcinoma, again, with the hope that these can be mobilized to attack tumors better. Okay. So there was a major paper recently in cholangiocarcinoma telling us more about the different types of connective tissue cells, which I think, again, will be some other cell type to understand how it communicates with growing tumors, as well as an emerging set of tools that we can use to try to use model systems to predict new therapeutics, and I'll be talking about that briefly tomorrow. So new directions that I've seen that you're going to hear today. I think, what is the program resulting from these epigenetic alterations? And a related question that you heard one of the awardees on, can we target epigenetic factors themselves so that aren't mutated? We need to learn more about immune evasion. We're going to hear about that at this meeting. There's Jesus Benales is very interested, and others, about metabolic reprogramming. Can we exploit the need for different nutrients in these tumors? Metastasis, and I think where we'd like to all get in the future is to understand more about the earliest stages so that interventions can happen much, much earlier. And I think that there's going to be some talks on that. The final thing I just wanted to leave is that I think one of the mandates of the Cholangiocarcinoma Foundation was to have a growing research community. And so I just put from PubMed, which is basically a compendium of all the research publications. We see a nice steady rise in papers here. I will say that it's dominated, and I think that's a good thing, by clinical advances. So this is a field that clinical, wonderful translational work has led to real benefits for patients. There's far fewer basic laboratory studies in this group, but in fact, and hopefully some of the next generation therapies will be informed by that research, but hopefully these fields will go hand in hand. And just as a kind of, we always have a control in science, so how does this look like from other cancers? Well, it's comparable. Here's glioma, but I think there's much more funding for glioma research, as one example. It's much more in the public consciousness, but I think that this shows the research enterprise. I think Juan Vallée referred to the challenges that he encountered early on in drug development, but I think that this is now overcome. More people want to do this research. They approach our laboratories. They choose it as their clinical specialty, and I think this meeting and this community has catalyzed this a great deal, and I think the publications are one indicator of that. So with that, I apologize for going over, and I look forward to talking to you further in the meeting.